So, hi everyone. I hope you're enjoying the class so far. We're in week 2. So today we're just gonna briefly go over everything in the chapter. We're gonna be going over protein misfolding, enzymes, and protein regulation. So let's begin. So we're gonna begin with protein misfolding, and a lot of this is gonna go over what Laura just talked about today. But hopefully it's still good review. So most proteins, right? We just talked about this. Like most proteins are held together by non-covalent interactions. So those would be your hydrophobic interactions, hydrogen bonding, and the electrostatic interactions. And so if you have high or extreme pH or salts, these can disrupt charge interactions. So those ions will start disrupting the amino acid R groups that are charged or are polar. And so this is going to affect hydrogen bonding and stuff like electrostatic interactions. And then interestingly, UV light can affect both covalent and non-covalent bonds. And so, right, light is essentially energy. And the stronger your light energy is, the more it can affect your protein. Sometimes what will happen is if the light is strong enough, it can just cleave the peptide bonds and your protein will start to break apart. And so why is this a problem, right? So misfolded proteins are actually a very disastrous consequence in the cell. As you can see in the graphic in the lower right? A lot of the neurodegenerative diseases arise from misfolding of proteins. And these misfolded proteins tend to aggregate, and the aggregates, let me get to the next slide, right? Aggregates are thermodynamically stable, right? So proteins, when we talk about protein folding, right, what drives protein folding is thermodynamics. You're going from a high energy to a low energy state. So generally the native conformation of a protein is one of the lower energy states. And so, we go towards lower energy, we fold towards lower energy, but along the way, there's still intermediates and alternate shapes that proteins can still take that are, thermodynamically stable. Maybe not as stable as the native conformation, but still stable. And so what the cell does is it has these proteins called chaperones. And these chaperones basically, surround the protein, and it provides a safe folding environment that helps correct misfolds and helps prevent stuff like aggregates. Because aggregates, what happens is when the protein unfolds, you expose all those hydrophobic domains that you buried in the hydrophobic core. And so these proteins, these sections essentially don't want to be exposed in the solvent. So what happens is you'll have just these like strands of proteins, and they start aggregating in like a planar sheet. Their hydrophobic cores basically just bind to one another, and they form these thick aggregates that aren't easily destroyed. And this is what leads to like Parkinson's and Alzheimer's. And so you can tell the cell really wants to not deal with that. So that's why cells produce chaperones. It helps reduce misfolds. And then, but sometimes like the chaperones can't do enough, right? Sometimes proteins, the amount of unfolded proteins is simply too high. And when this occurs, the cell goes into heat shock response, at least in the nucleus and the cytosol, right? And so what it does is some misfolds are targeted by ubiquitin-based proteolysis. So you don't know what this is. It's when a protein is targeted with a ubiquitin tag. Like ubiquitin, it's its own protein. So that gets tagged onto whatever protein the cell wants to destroy. And then the ubiquitin acts as a marker for these proteasomes in the cell. I think Laura talked about it just today. So proteasomes are just big multi-complex proteins that can take other proteins and break them down into amino acids again. And so this reduces the number of misfolded, unfolded proteins in the cell. And another thing is, right, upregulation of chaperones. This is just to assist in folding. If you look in the comic in the lower left, right, you see the oval shape is a correctly folded protein, and then it becomes spiked or misfolded. And the misfolds aggregate. So the cells either tag the purple ubiquitin and then it's destroyed, or it's helped by a chaperone. And so that was in the cytosol and the nucleus. In the ER, there's a different response. It's called the unfolded protein response. And so it's a lot of the same concepts as the heat shock response, right? It's the upregulation of chaperones. And then there's no proteasomes in the ER. So in the UPR, what the cell has to do, it has to retrotranslocate proteins out of the ER. And when it leaves the ER, the proteins are tagged with ubiquitin, and then it can meet proteasomes in the cytosol to be destroyed. Then the third thing that happens is an upregulation of lipid synthesis. So why the cell does this? Well, the ER is composed of phospholipids, right? So if you increase lipid synthesis, you're trying to increase the volume of the ER. And this makes sense, right? Because you're increasing the chaperones. You're creating all these other proteins, right? The UPR, there's a lot of proteins that have to be created to initiate this response. So you want to increase the size of the ER, basically your workspace. And so, anyways, with that aside, we're going to move on to protein ligand interactions, affinity, stuff like that. And so let's just start with what are ligands? So ligands is essentially anything that binds to a protein. Sometimes ligands are other proteins, sometimes they're vitamins, sometimes they're ions, a lot of different things that this proteins bind to. Anything is a ligand. And so how do proteins bind to ligands? Essentially, they leverage different amino acid properties to form weak non-covalent interactions. And it's really the accumulation of many, many interactions that allow for binding to occur. And if you look at the graphic on the right, so you'll see something like serine. So like serine is polar, right? So it can form hydrogen bonds with the ligand, or something like alanine is non-polar. So it would form hydrophobic interactions with the ligand. And because this utilizes R groups, it's orientation specific. Like again, if we look at the diagram on the bottom right, you can't just flip cyclic AMP. Like if you took that molecule and you flipped it around, the same interactions can't occur in the binding pocket. So we would say protein ligand interactions are orientation specific. And not all interactions have binding pockets. Some have flat interfaces. And what really is important to remember is most interactions are random in the sense that in your cell, right, you have the cytosol and you have proteins floating around in that solution. How do things react? Like any reaction, you require physical interaction. So the two proteins, or the ligand and the protein, need to physically hit each other. And so this is the fusion base. It's random. And there are ways the cell gets around this, but we'll talk about that in a later section. And so let's just do a quick question. Let's simulate the problem set. So if the glutamic acid, so in the bottom, it's like this green color right here. If the glutamic acid was replaced by aspartic acid, would the ligand still be able to bind to the protein? And I included the aspartate. The R group is to the right of the nitrogen if you're confused. So you can write your answers in the chat, or you can just speak up. Anyone? Well, I mean, it's fine. We can just talk about it, I guess. So I guess the first question we should ask ourselves in these types of questions are the two amino acids similar, right? Are their R group chemistry similar? So if we look, the glutamic acid, what is it doing? It's creating a hydrogen bond between the carbonyl oxygen and the hydrogen off the oxygen from cyclic AMP, right? So if we look at aspartate, could it create the same interactions with its R group? And the answer is yes, basically, right? Like if you look at the two, they're very similar in their R group chemistry. And so, you know, if you actually did it in real life, it might not work. You can never be sure about these things, right? But because their R group chemistry is similar, there's a good chance that it could work and binding would still occur. And they also share the negative charge, right? So if we look at the oxygen, right, one of their oxygens is negative. So it's a charged amino acid. They're both charged amino acids. So, you know, very similar chemistry, maybe, probably. But if we ask, what if it was replaced with arginine, right? So arginine is the yellow compound. So if we look at it, sure, right? Nitrogen can form hydrogen bonds, so they're similar in that respect to their R group chemistry. But a big difference between glutamic acid and arginine is that glutamic acid is negatively charged while arginine is positively charged. And so switching the charge is probably not a good idea, especially if it's an ionic bond, right? If you switch the charge, it won't work. So arginine is less likely than aspartate. It probably wouldn't be able to bind if you swapped glutamic acid for arginine. So, you know, this is the kind of reasoning you want to have when you're answering the problem sets. All right, moving on. So the next key term we'll talk about is affinity. So affinity is just the strength of the protein ligand interaction. And there's essentially two factors to this. It's the number of interactions between the ligand and the protein, and also the strength of the interactions, right? And so I think we talked about this again in class. So most protein ligand interactions, right, are non-covalent interactions. So they're not the strongest interactions, but cumulatively with all of the interactions combined, it can create a strong enough interaction that they will bind together. And so, yeah. And then we have the Goldilocks hypothesis, trademarked by Laura Francis. So the idea is essentially most proteins want to have a moderate affinity for ligands. And this is because having a, right, if you think about it, if you have a too low affinity for your ligand, what's the point, right? If you can't even bind to what you're supposed to bind to, then is it really a ligand? But on the other side, right, you can't have too high of an affinity either, because most protein interactions, they're non-covalent for a reason. They're not meant to last forever. The problem is if you have too high of an affinity, right, you'll never let go of your ligand. And if, right, most reactions aren't infinite, they occur and then you let go. So if you bind but you can't let go, then essentially your product is not free to do what it needs to do. And so that's the Goldilocks hypothesis. So at this point, we're going to talk about KD. And so KD is the dissociation constant. In like a general sense, it's the rate of dissociation of a protein ligand complex. So the greater the KD, the more likely it is to dissociate, or the faster it is to dissociate. And so if we look at the equation, KD is equal to the concentration of free protein multiplied by the concentration of free ligand divided by the concentration of protein ligand complex. And the physical interpretation is essentially KD is the ligand concentration at half saturation of protein. So if we look onto the right, if we assume that the protein concentration is equal to the protein ligand complex, so basically, right, half of your proteins aren't bound while half of your proteins are bound. That's what it means to be at half saturation. If that's the case, we can basically cross out P and PL. And all you're left with is KD is equal to the concentration of ligand. And so it's just key to remind yourself that KD has an inverse relationship to a affinity. Like the higher your KD, the lower your affinity. You don't want to mix that up on your problem sets. So let's do a quick check-in. So number one, why is too low or too high affinity for ligands detrimental to proteins?
If it's too low, then it won't bind to the protein. But if it's too high, then it won't let go of it to like go do its job again.
Yeah, that's a great answer. And so number two, why are protein ligand interactions primarily composed of non-covalent interactions rather than covalent interactions?
They're non-covalent. That way the affinity is not too high and it can let go of it again.
Yeah, basically. And the problem with covalent, like it takes too much energy to destroy or form them and destroy them. So it's better to just not form covalent interactions or any protein ligand interactions. That way they're fast. And so that's the end of my section. I'm just gonna turn it over to Gabe.
All right, can you, do you have one more question on the last slide?
It's the same question. It's fine.
Same question. Okay. All right. So for my section, we'll be talking more about enzymes. So I'll start by talking about why cells need enzymes. So as you can see on the bottom here, it's kind of a generic picture of enzymes. Enzymes are catalysts and they carry out chemical reactions. They make and break covalent bonds. And they also are in charge of the formation and destruction of covalent bonds, and those actions are chemical reactions. And so ribosomes form the covalent bonds between amino acids. That's what's linking together the chain. That's one example of an enzyme. Enzymes, the way they do this is they change the activation energy and increase the frequency of chemical reactions, which we'll go into a little more in depth. Enzymes can also be easily regulated, which is really important. They can be like turned on and off. And not all enzymes are made of pure 100% protein. Some enzymes are made of RNA and protein mixture, where RNA is the catalytic molecule that helps like catalyze reactions. These are called ribozymes or RNA enzymes. We go to the next slide. Okay, so here you can see that this is a picture of the reaction process diagram. And so at the beginning, it shows the reactants and its products. And the way that enzymes speed up chemical reactions is they make the transition more energetically favorable. As you can see, it takes a certain amount of, takes a lot of activation energy for many reactions to occur. And so what they do is they reduce the activation energy by changing the microenvironment around the substrate. And can you go to the next slide? Here we've got more of an explanation you guys need. So the diagram itself tracks reaction progress over time and shows free energy associated with the reaction. The free energy is on the y-axis and time is on the x-axis. And so there's always going to be like a transition state that the reactants have to go through to become the product of reaction. The transition state is too unstable though, and which is why it's associated with the higher energy state. And so the activation energy is a difference between the transition state energy of the products and the transition state and the reactants. The enzymes lower the activation energy, as I said, and they stabilize the transition state to speed up the reaction. And you guys can just read the rest of it. It's basically what I just said. Can you go to the next slide? So now the big question is, how do enzymes change the microenvironment to lower the activation energy? And so there's three main ways this can happen. Enzymes bind to two substrate molecules and orient them precisely to encourage a reaction between them. And so by placing them close, obviously it's going to help a lot more. Like Robert was saying, if they're all over the place, they can't physically run into each other, and they need to, be together. The second way is binding of substrate to an enzyme rearranges electrons in the substrate, creating partial negative and positive charges that favor a reaction. And the third way is C, where enzyme strains the bound substrate molecule, forcing it towards a transition state to favor a reaction. So now moving on to the regulation of protein function. So one of the ways that proteins can be, or one term that we'll use a lot in this class to describe regulation of proteins is allostery. And so basically the main idea of allostery is that structure equals function. And so there's many different ways that this can happen, but basically a regulatory molecule binds to particular places on an enzyme, which either can cause it to activate by like opening up the active site, or it can cause it to close the active site, or as you can see here, other sorts of like formations of active sites, or causing it so that it's not able to the distortion there, so its substrates can't get in. So there's a variety of ways that they can be controlled, but the main idea is that a molecule is coming in, causing a change of shape, which causes a change in the enzyme's function. So again, structure and function is the main idea of allostery. All right, here, next one. All right, so post-translational modification, this takes place typically after. So proteins are often regulated by chemically modifying covalently the protein after it has been made and folded. And so these modifications are typically they're reversible, though in many cases the modification is never removed. Post-translational modification is often used as a switch mechanism to control a protein's function, to change its surface protein or protein's surface chemistry, to change its structure, change its function, and be able to reverse that by removing the modification. And so one big example that we'll talk about time and time again in this class is going to be kinases and phosphatases. So kinases are a group of enzymes that they place phosphates onto proteins, and sometimes to activate, sometimes to disactivate. And then other times, phosphatases will then take off the phosphates, which can either activate or deactivate proteins. And so this switch mechanism is really key in many proteins, where, like I said, the kinase can put the phosphate on, phosphatases can take them off, and this can continuously happen like in a cycle, like a switch. There, as you can see on the diagram on the side here, this modifications can be made in many places along a particular protein in different ways. And so this creates, this like mix and match creates the power of combinatorics, which is the different combinations of different modifications, which allow proteins to act. So for example, I'll give the example of P53. Using the power of combinatorics, the different modifications allow P53 to act as a central coordinator of cell stress response, but in many different pathways, depending on what combination of chemical functional groups are being added to the protein itself. All right, you go to the next one. Okay, so nucleotide binding and hydrolysis. So we'll be talking about many different types of GTPases and ATPases, but basically the way that these proteins are regulated is through being attached to GTP or GDP. And so these categories of proteins are enzymes that they bind, like I said, to GDP or ATP, but they have the intrinsic ability to cleave the bond between the last phosphate. So each one is like, GTP is guanine triphosphate, and then GDP is diphosphate. And so there's 3 phosphate groups. And so these enzymes have the ability to cleave off the third phosphate. And they have other proteins that will help, but basically when they are attached to GTP, sorry, they are active. And then when they do the hydrolysis, then they are inactivated and are like attached to GDP. So this like allows them to be like an on and off switch, where, like I said, when they're attached to GTP, they're switched on, and then GDP switched off. And same thing goes with ATPases. ATP, when they're bound to ATP, they'll be on, and then when they're bound to ADP, they'll be off. And this is just an example at the bottom of RAS, which you guys will learn about soon, which is the GTPase. All right, so the next slide.
I think that's the end.
Is that the end?
Yeah.
Oh, you're right. Yeah. My bad. I thought there was one last one. Any questions? On the content, and then we can move on to problem set questions after the recording's over.
All right. If there's no questions, I'll just stop recording.